All right. Hello, everyone. I am super happy to introduce you to my startup, Delineate. At Delineate, we are focused on applying large language models, very specifically AI agents, which are a kind of complex system of specialized large language models, which I can get into a little bit more later. But the goal is to help pharma companies and biotechs design better clinical trials. So by way of introduction, I am Emily Nieves, excited to network with you all. I'm co-founder and CEO of Delineate. Before this, I was actually a CSAIL PhD student. I was in the Julia lab. And before that, I worked at Pfizer and AstraZeneca in their clinical development division. So I consider myself a scientist / engineer. I'm kind of on the side, in the middle. And my co-founder and CTO is Jawad Iqbal, who was involved in some really transformative digital transformation and AI solutions at Lockheed Martin. So a lot of similarities in the defense industry and pharma, so he brings a lot of great experience there. And we're super grateful for all the support we've received from MIT and all of our investors. So we actually started out as part of the MIT Sandbox, which gives some funding and lets you explore your idea in a lot of great mentorship. And then we actually recently finished up Y Combinator out in San Francisco. And then we just finished raising our seed round from these great investors on the side. So really grateful for all the support we've received. So the problem that Delineate was started to solve is the problem of bad trial design, which can lead to failed clinical trials. There's a lot of buzz on AI drug discovery and finding the right molecule, finding the right protein, but that's really only the first piece of the story. It continues. It's really difficult to predict what will happen when you give a small molecule or a protein to humans for the very first time, and that's something that we help companies with. The headline here is, bad trial design leads to failed trials. So if you choose the wrong dose, the wrong patient population, an entire clinical trial can fail, and you can, lose out on all the scientific work and money that you've put into it, even if the drug was, quote unquote, correctly designed. So, one example is if you give Tylenol at too high of a dose, you would fail phase 1. You would see toxicity issues. And then another, kind of core example that we all think about is the Keytruda case study. I don't know how many of you are familiar with that, but it's kind of like the blockbuster oncology drug. And Merck did an amazing job at picking a very specific patient population. So they saw an amazing outcome when they did their trials. And they were head-to-head competing with all of the other top pharmas in this space. They won out. Keytruda is half their revenue, and all the other companies kind of failed to make headway in that space. So designing the right trial and picking the right parameters, which looks like dose schedule, and the right patients, can mean the difference between a success and a failure. And so we try to help companies by helping them build the computational models that can predict the trial outputs and help optimize those parameters. So in principle, these pharma companies do this already. They have teams of very smart scientists doing this. It's a combination of a lot of pharmacology, it's math, it's computer science, and it's data science. And they're really hard to do. So I worked building these models at Pfizer and AstraZeneca. One model I spent 3 years of my time building, which is far, far too slow for those clinical development pipelines. So the problem is that we can't always use these computational models because they take too long to build. So sometimes the clinical teams move on without us. So we are spinning our wheels trying to build these amazing simulations, and they're kind of going forward with the best dose guess that they have using competitor data, et cetera. So really, Delineate wants and envisions the future of drug development, which is data-driven. There is a perfect model for every clinical question, and we can move forward with the best decision possible. So how do we do this? So we apply, like I said, AI agents, LLMs, good old-fashioned software engineering to 100x the speed of computational model development to help inform these successful trials. And so this has kind of 3 pillars is how I think about it. So we're replicating the scientist's workflow and helping them along the process and taking a lot of the tediousness out of what they're doing. So the first pillar is finding relevant studies. So we use a ton of literature data, both in competitors' clinical trials, but also publications that cover biological mechanisms and disease biology. So we have our specialized LLM-based search that finds the relevant studies, make sure it has the data, make sure it matches the criteria for the model that you're trying to build. And then once we find those hundreds or thousands of PDFs, we have to structure that unstructured data. So we go from just PDF and data and graphs to actually something structured. So building data sets that are ready for training an AI model, for building a simulation. And then once we have that, then our AI agents, they actually go through and perform the analysis. So we assist with building that perfect simulation, that statistical model over top all of that data that we've constructed. So the first pillar, as I mentioned, is finding and then extracting data from existing research. So this is usually tons of research papers. One key feature we have is extracting data from clinical trial publications. So we can basically recreate the data that went behind the paper that's not usually released with the paper because it's all competitor data. But we can actually basically reconstruct everything that happened in a clinical trial from a publication. And so another thing that I'm super proud of is we actually built our own computer vision model to extract precise numerical data from graphs. So that's a kind of core problem in industry. We use like these digital rulers. So we're like PhD scientists measuring points on a graph. And some scientists can spend weeks out of the year doing that. So we've actually automated that. So that's a kind of pillar of our technology. And then, as I mentioned, we also have AI agents that help build the computational models that go alongside this data. So it has all of the information in context on what that data represents, and so it can use that information to build the best model. So this is actually an example of something called a quantitative systems pharmacology model, which is basically a mathematical model simulating side effect of a cancer CAR T cell therapy drug. So we have a lot of data in context and information on mechanisms of action, and it's actually writing up the code to simulate that using best practices in the field. And we fine-tuned our embedding model and our agent system to kind of align with those best practices. And so this agent-assisted creation of these predictive models is scientist in the loop. So the scientist is helping by way of chat interface to design the best possible model and iteratively improve it. And then this would, of course, be used to predict the trial outputs, and then you can see what happens when you try a different dose, try a different dosing schedule, et cetera. And that helps companies optimize their trial. And so we're super proud of the work that we've done with some of our pharma partners. So with Sanofi, we've actually recently were acknowledged in a publication from them, which we were grateful for. So we actually helped them build the first of its kind small molecules database for training an AI model. So this predicts the output of a phase 1 and helps you optimize that based on compound structure. And so we processed over 1,000 publications going back 30 years, digitized a whole bunch of pharmacokinetics data, et cetera, and created this really fit-for-purpose data set. So once we delivered it to them, it was ready for their model training, and they were able to train in a couple of weeks and deploy that model in-house. So we saved their scientists a couple of months of effort of putting together this data set. Similarly, with Roche, we have helped them conduct one of the largest meta-analysis of its kind. It's a particular statistical meta-analysis that essentially condenses all of the competitor information and then helps them make a decision on whether or not to move forward with the next phase of their trial. And so here we leveraged our agents and our extraction techniques to normalize data across all of these studies and create these fit-for-purpose data sets that then they were able to plug their internal data into and run their models. And so this one is not public information yet, but we are also working with another large pharma company to essentially deploy our AI co-pilots for their scientists to use to develop these models. So as I mentioned, these models can be AI, they can be traditional statistical models, or kind of more simulation style. And so the system of AI agents works with the scientists. It's iterative, so it tries to fit the data, it tries to match the behavior that we're seeing, and it's trying to capture all of the relevant biology, and it works with the scientists to make sure it's capturing that accurately in a loop. Yeah, I talked a little fast, so I have a couple more minutes left. But at the end, just wanted to say, would love to collaborate with anyone, even if you're not in the biopharma space. We've built out a really great, complex document processing engine, as we call it, and if you have needs related to that, would love to speak to you. Also, we're hiring, so if you know of any great scientists and engineers in the AI biospace, feel free to pass the resume. Would really appreciate it. Feel free to email me or catch up with me at the networking sessions, etc.